So I suggest that each group should appoint a group leader and then the members. So let's go now to the format. You don't need to write or prepare a very long lab report. It should be concise lang, but substantial. Okay? So here, measurement, analytical chem is entirely about measurement. What these measurements signify and the understanding concerning the quality of these measurements. So with this, you will be also applying statistical tools in expressing your results. So that would mean more than 2 replicates. So at least 3 trials for you to apply your statistical tools. Okay? So in this class, you will be performing a number of experiments to measure the concentrations of various analytes. Again, class, analytes are the ones, the substance or the compounds that you will be testing, no? And performing okay. And the results, you will apply your statistical analysis and be able also to interpret based on whatever references you may have in the internet. So, hindi lang pwede nga may result na ka mo. What will you do with your result? Kung di ka mo kabalo mag-interpret, no? So, I would need also interpretation. Okay. Quality of measurements. In each experiment, you will obtain sets of data. So, as I've said, at least 3 replicates. This data will be statistically analyzed to measure the quality of the data, and accuracy and precision will be determined. Okay. So this will be the determining factors to judge, if the quality of your measurements are reliable or not. Statistical analysis allows you to mathematically determine a higher relative degree of accuracy and precision contained within your data. Okay. You should also understand the concepts behind what each applied statistic model reveals. So you should have good. How long will you resource? Ini like this one. Okay, so as it is good statistical analysis, that means also included in your statistical results are interpretation. And this is one fundamental key to sound scientific ability. So remember, class, and definitions ng chemist. No? A chemist is a scientist. So you must learn to become one. Kaya diba later, future chemist na mo when you pass the board exam. It needs a lot of patience and perseverance. Okay? This one, I think you are already familiar with this, no? Since senior high. The difference between accuracy and precision. And this will be part of your computation. So accuracy is how close the mean or the average of all your data points to the actual or known or true value. It reflects the degree of systematic error in your experiment. While precision is related to repeatability or standard deviation, diba? It is a measure of the variability of all data points, the degree of consistency, and reproducing. So reproducibility also, the term, no? The same data points. It reflects the degree of random error in your experiment. So take note, later in our lectures sa Analytical, we'll be clearly differentiating systematic error and random error. what are those? No, and accuracy is often it often reflects the degree of systematic error, while precision um, reflects the degree of random error in your analysis. So in your during the performance of the test. So this will also challenge your skills, no? In like example, pipetting, in delivering liquids, and how you perform the entire test. So makita na sa mga errors. Okay, now going to back to the report format. So of course, you're required to place the title of the experiment or the lab activity, then the leader and the members. And since this is a group effort, so parehas ang grades syempre per group. Kaya naman pwedeng isa man lang ka-lab report. So expect that uh, the the points or the grades per report with only one, and that all the group members would also have the same grade. Okay. Sa report lang plus ha, but lain naman ang attendance syempre and also participation. Okay. So that the report consists of the title, the abstract, introduction, experimental procedure, the results, of course, then discussion of the results, then your references and appendix. Appendix includes like references, like standards, or your tables, or your computation, or your pictures, no, during the lab activity. Okay, so for abstract, these are the reminders. No, you include what has been performed in the lab, just a summary of the summary, summary of results and conclusions. Report what you did and what you observed. Should be brief. Maximum of 3 sentences, but long sentences would be okay. Next, and this is an example of a good abstract. A single solution of unknown chlorides from sodium chloride in water was aliquoted and titrated with silver nitrate through Fajan's method. So this is what has been performed in the lab. It can be said that 95%, so you already have your statistics here, that the true mean of the percent weight of the chlorides from the titrated sample was 59 points. So take note also of the significant figures. Now plus minus 0.2479%. So this is your standard deviation, which is also related to what? Precision, you know, and the here random errors, diba? Precision. Okay. So your abstract should be very specific. It should contain or include what was done, what was discovered. So what was discovered, this is your result. Very brief, take note. Para hindi ko yaman check. So it could be 2 to 3 sentences, okay? And take note also of the tense, no? Okay, what is an example of a bad abstract here? Again, repeat siya ang objective. This is not necessary. The main objective of this lab was to determine an unknown chloride ion concentration with the process of titration. We know titration is, di ba nga, i-include pa din, with the known concentration of silver solution. The lab performed the standardization. Of course, standardization, nag-titrate ka ganit, di ba? Dapat mas standardized. It should not be included in the abstract. It's part of the methods, no? Methodology or procedure. The lab performs standardization using Fajan's method with adsorption indicator that was dichlorofluoro reasoning as indicator. So where is the result here? Wala. No? Okay, so never start an abstract by describing the objective of the experiment. Take note of these group leaders. Say what you did and what you found. So you have your results. This does not specify what was observed or discovered, this one. And the indicator is a detail which belongs to the experimental section or the procedure. So practice. Okay? Practice na kamo. Okay, introduction. Address the experiment or the problem being investigated. So, pwede ka mo din magbutan sa objectives. Answer the significance of the task at hand. Address the significance of the task at hand. Discuss the concepts. So, you could also research on the concepts, the principles, the theory, on what the experiment is anchored in, no? Involved in the experiment. So, dire ini sa introduction. If you choose to discuss the history of the chemistry applications, this is where you do it sa introduction. But it should also be like 3 paragraphs. No, okay na na. Or 2. So I hope you have also read this one. Like for example, a good introduction. A Beer's law plot, magaling ang Beer's law. So this implies that you have done, ito lang graph to ginpakita ko sa inyo with calibration graph nga may standard. So it's Beer's law. So a Beer's law plot and a standard addition plot are analyzed in this experiment. So these two methods are used to determine the concentration o si ari ng objective nino to determine the concentration of a known solution. The latter plot, unlike the former one, considers the matrix effects. Ano gani ang matrix? You remember sa lecture to yesterday? Now, it is in where the analyte is embedded. So whereby the wavelengths of light emitted, so and so forth. Then, diba? But class, don't use first dapat third person. Amo na siya subong so dapat avoid using we, you, yun, amo na siya, or I, no? So we can how do you praise this one? Okay. Without using the first person, dapat third person. Okay, next, according to Beer's law, the absorption of the species of interest is directly proportional to the concentration of the species and so forth. So the objective of the lab is pointed out. The theory behind to what you did, no? So again, refrain here from using first person. So we have here 6.05 ml. Pero sa thesis, balag yun na suggest nila nga do not start your statement with numbers. Sa thesis na siya. Okay. Next. Okay, what about results? So here, good results section include finalized and tabulated results that are not already in the Excel spreadsheet. So in the pwede class nga, ang Excel nito ging screenshot nyo lang yung butang sa sa portion sa result. Dapat, you must make a table with Jaya. Next, cite the Excel file for all other data. So if you have other data nga daw lagrawig, so you cite na lang the Excel file. Next, include finalized graphs, charts with proper formatting, titles, labels, no? Next, make sure the graphs are large enough so it can be clearly seen, especially the data points. Next, include any detailed data calculation. And if there are some errors in the test that you repeated, so i-include nyo man. Next, perform long calculations in the appendix. Itong, like, for example, gin-check nyo ang regression sa calibration graph nyo, tapos gin-manual nyo, wala. But hindi naman subong kinalan nga manual kapag hindi mo mga calculators naman and functions sa Excel. Next, bad result. Do not copy and paste the same information from your Excel spreadsheet into your formal written report. Do not forget to cite all other data results that you have been accrued in your Excel file, and do not list and answer the question asked in the experiment. So, okay. Okay, what is missing in this graph? Wala sang title, wala sang legend, wala sang data, wala sang title ang ano ni siya, nabutan ko ano ni siya ang parameter ang y-axis, and same with the x-axis, puro lang numbers. So this graph has no title, no axis labels, no units also. The numbers are so small, no? So wala man sang descriptive factors. And wala man legend. Okay? So this graph would not get a good score. Okay, this is what should be in a graph, no? That you want to contain in your results sa report. Okay? So, amo ni siya ang mga table. Good result sample. This person listed only data which was not tabulated already in Excel. Baka summary lang ba? Person also cited where you can find appropriately formatted and organized data calculation. So it's in here. From data, okay. Then see appropriate Excel sheets for graphs, calculations, and other tabulated data. So remember to always put units with all of your raw uncalculated data. Diba, ang chemist should have perseverance. So in both, dapat may pasensya in both your formal report and corresponding Excel template. So dapat may units. Okay, this is the most interesting part of your report. This is in where you get to interpret data, discuss your calculated results, and what they mean. Ano ang applications ng inyong result? Okay, we're used to, di ba, sa unang mag-experiment. Okay, ada ng result na dayon. Ano mong result, no? So, what you're doing, no, what your results is all about, and would find more meaning if you know what they mean, you know how to discuss it, you know how to interpret it, no? Do not list or answer the questions in number format. The Excel templates have sections for each of the questions in the experiment. You are supposed to solve, calculate, answer the questions in a template. Okay, so here, Write in third person. Scientific experiments demonstrate facts that do not depend on the observer. Therefore, reports should avoid using the first and the second person like I, me, my, we, our, or us. Using the correct verb tense also, lab reports and research papers should be mainly written in the present tense. We should limit the use of the past tense except for sa mga procedures, di ba? So use past tense to describe specific experimental methods and mga past observations and inciting results published in the past. So take note of this, ha? Kay, di ba, later, mathesis na kamo. So this could be a step, no? Do practice lang to what you will be doing when you are fourth year. So this is a bad discussion sample, no? Numbers, diba? Dapat hindi number format. Next, discuss the implications of your data in light of the questions asked. Dapat paragraph format. What else? Discuss areas of possible improvement or different types of error which might have occurred during the experiment. So, be observant. Ano ang possibility nga may mga slight errors? And I would require you to also list the random and the systematic errors that might be affecting the results also during the course of the experiment. So this is how discussion should be. This result suggests that the concentration of sodium and, okay, sama ni siya, sahaw na lang. Next, interpreting and discussion of the implications of your data is the primary point to all these experiments. So, ano na siya nga nag-experiment ka muna? Ano ang result ng 7 point, sorry, ano ang result ng 9.0 sa water if it's drinking? Ano na siya ang meaning? Aside from being highly or very alkaline, ano na siya ang meaning? Is it potable for drinking? So, gaano man importance, no, or meaning ang result, ano na ang application sa like, nag-analyze ka mo sa water from refilling stations. So, you must be able to find out, no, if the results are fit for drinking, based on what standard. Na para at least maanan ka mo na how to have a good discussion. You go extra mile. Okay? Next, appendix references. Cite any references used. Okay? Next, use your appendix to type in long calculations and error analysis which you did not or could not perform in Excel. Remember to cite this in your result section. So during submission of result, I would prefer in word format para ma-edit ko and masulatan ba yun ang grades or wala dasig rather than in PDF. So, submit results in word format. Okay? Next, here. We'll be discussing this sa Analytical. But I'm sure you already know how to compute standard deviations and confidence interval. Okay, now I will
and also there's a label, date received, the sign of the one who received, and the date opened, and also the name of the person who opened the chemical, and also the expiry date. This is the ISO standard of labeling the chemicals. And also, most important thing sa laboratory is the fire extinguisher. First one, and also before you enter the laboratory, you must have the fire extinguisher. And this area is for drinking water analysis. Dire niya siya ang physicochemical with 11 parameters nga requires ang DOH. And also, the reagent bottles should be labeled with the name of the reagent, the analysis to be conducted sa ammonia reagent, and date prepared, name of the person who prepared the chemicals, and also the expiry date. And also, you can see the GHS of the chemical. Okay, in this area, you can see the uploading balance, and also this is also a spectrophotometer, but ancient na, dogay na siya. And this is a steering hot plate, and this is a refractometer. The refractometer. Okay, this one is the BOD ref, where you restore the samples for biological oxygen demand, and it is incubated for 5 days. And mabalik ta sa spectrophotometer. Amo na siyang set up. Okay, touch screen siya, guys.
Checking main features has been completed approximately. Yeah, the sample can be measured. Select a measurement mode.
Okay, first, you can choose a system, ATC, single wave, or you can choose multi-wave.
ATC mode. ATC mode leads to measure absorption, transmittance, and concentration before measuring. Input wavelength and select cell holder at setup menu.
Have you heard the set up wavelength?
Set up the measuring
Okay.
conditions. After
Here
setting
one.
up each item, touch apply button.
Wavelength is depending on the sample you want to read. For example, for the phosphates, the wavelength is 690 nanometer. Just like that. And this is the compartment. Okay. The compartment is rotating, and this is where you put the cuvette. Wala na siya blank, guys. Direct na siya nga, one compartment lang for the sample. But SOP, you should read the blank sample. Because sa dere, sa piyak, sa aton nga UV vis na Shimadzu, mayroon siya, amo na siyang compartment different. This compartment is for the blank sample, and this compartment is for your sample compartment. Okay, there's a difference in their compartments. Okay. Next, in this area is the preparation area for the metal analysis. And this is Miss Antoniela, the one who analyzes the metals using BAS, or atomic absorption spectrophotometer. Isang area sang
Si Miss Anton Reg ang metallic Metallic.
Ames.
Si Miss Anton
Okay.
ang metallic queen.
Yes.
Queen of metals.
Queen of Metals. Okay. Okay, si Miss Anton is reading subong. You can see. Okay. Hindi na siya ma-appear ang readings ni Miss Anton. And also, it uses three different gases: the argon, the acetylene, and the N2O. And also, it should have a exhaust because AS is using flame. As you can see, that blue one or that one red, it's a flame. Okay. In this area, we analyze food analysis, food samples, because we are already accepting food samples. And ng Feresa is the one who analyzes all the parameters for nutritional facts. You can see the samples. Okay, this area is for the TSS area, total soluble solids, total suspended solids. And also the hot plate or magnetic stirrer. And also we have cabinets for volumetric flask from 250 ml to 2 liters. Also another cabinets for different kinds of chemicals sorted in their compatibility. And also the burette used for titration. And this area is for the wastewater analysis area. This is area that we analyze the biological oxygen demand. Okay. And also, we have the HPLC room or high performance liquid chromatography area. Okay. This is the HPLC. And also we have sonicator. Okay. Next. Also, we have the digestion room where we digest our metal samples. And also, we have rotovap for extraction. Also important inside the laboratory should have emergency shower in case of accident or something nag-spill sa mga body, and also the eye wash, important. Also the PPE cabinet later. Also, Last part is the oil and grease area. Meron siya sa own nga fume hood. And this is the area of oil and grease. Also the glass spares. And also it should have an emergency exit and then exhaust. A washing area. Last part is first aid cabinet. And also the PPE cabinet. Okay na siguro, miss?
Okay. Sige, sige. So, do you have any question to Sir Reggie, class? Any question? Anything that you would want to see? Wala? Angela? Yes? Reg, sa baba ko ni Reg ang PCR.
Okay. Kaya muna siyang micro lab. And also we have stairs nga may pakadto sa Aqua Lab. Aqua and PCR lab. PCR niya sa sa shrimp, hindi sa tao. Okay, this one is for Bacte for aquaculture. And this one is the PCR section.
Ano ka na-reg sa COVID section?
COVID section.
Sa mga shrimp.
Wala patient. Wala patient. Kamo na siyang sa babaw.
Analyze plus buong reg. Nagasiga ang suga sa piyak.
Okay.
Para.
Pero wala man din
siya ang master mix preparation area. Ang orange ni siya, micromix, microcentrifuge. Para. So instead of the usual na centrifuge sa CHEM, amo na siya ang sa molecular bio. So it's microcentrifuge. Okay na next step? Okay na rin. Pwede man to sila pipette. Pwede sila. Reg, hindi ka na makita. Sa PCR, nag-under ko rin. Sa second room. Sa second room na lang. Ano ko lang i-explain. No, sa piyak. Sa piyak. After sa piyak. Sa pinaka-last and the second one. I-open lang. Ang light, please. So these are the PCR machines, PCR, polymerase chain reactor machines. And this one is the rapid test. in just a matter of like, for the whole process, makuha na siya in 2 to 3 hours, including sample prep. Next, the other one, the red one, Reg. The red one. Okay, amo ni siya ang first na PCR namon. Conventional, we call this conventional PCR because before we are still after PCR, after this amplification, gakadto na dayon sa electrophoresis. So, ang PCR amplification is, and DNA amplification is just like photocopying small portions of the DNA into billion copies para at least it would be possible for the analyst to determine kung ano na siya ang, if it's a virus or not, no, by multiplying amplifying the DNAs. So it's like in forensics, gamay-gamay lang siya na amount of crime scene evidence, pwede na siya ma-PCR because the PCR functions as, kung sa layman, Xerox copier, ginadamo-damo yung copies ng DNA for the detection to be made possible. Amo na siya ang functions ng PCR. But of course, mayroon na siya conditions like temperature. So, i-set up na siya, may program na siya. Number of cycles per minute. So, okay na? Kita na ka mo PCR machine? Alright. We have 4 units actually in this room. Okay na, Reg? So that would be all for now, and I will just send to you your first experiment. It would be calibration of volumetric glasswares, and that would be next week. So,
Okay, Miss.
come again. What deadline? Hindi. Bali, during your laboratory activity, di ba, every group is required to generate a laboratory report based on what we have discussed, based on the format that we have discussed. So example, if the lab is Friday, I would expect that during the weekend, you would already pass the results. The report, I mean. So is that okay? Okay. One more thing, ano po ganyan nagpat ako. Okay. Be able also, na-access nyo na ang inyo na module. Nahatag na nyo siya. So, and be prompt also to answer your progress checks. And I will just post in your group messenger the deadline on when to submit. Okay? So during lab analysis, feel free to ask questions. Whoever is assigned, kay usually, yun na divide and this is staff mo kay actually, ang services naman to the academ is part, since we are a cooperative, it is part of our social responsibilities at this time, no, during the pandemic, na ma-extend kami sa assistance sa society. So, part na sa among social responsibility to share what we have in here, especially sa academ. That's why nag-okay ang among board of directors to have virtual laboratory activities. So, make the most of it. You can ask questions to whoever is assigned sa test. Okay?
Okay, Miss.
Yes, Miss.
Thank you, Miss.
Kuya, kada ba second year na kamo. So, wala man na sa, di gonna refrain nga, hindi mag-question. But be able to ask mga sensible questions, bara, nga that would also help others, especially your classmates. So, think of kung ano pa na ang mga questions nga would make you learn better. Kaya di ba virtual would like, unlike sa face-to-face nga makita niyo gid ang actual. So help each other, no? Ano kamo generate kamo sang ideas and questions that could also benefit your classmates. Okay?
Okay, Miss. Thank you, Miss.
Miss.
Okay, so see you again next week.
Thank you, Miss.
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 Recording